So I will present today a use case where I used Knime to FAIRify some data. And Patrick was talking about it already, and also Michael, but I also assume most of you know it, but just to bring everyone on the same page, what is FAIR? So FAIR is an acronym and stands for findable, accessible, interoperable, and reusable. And there are a set of principles that have been coined in 2016 that are focusing on making research data and objects available, but also most of all reusable. So in order to have data reusable, the first step is to find the data, and therefore it needs to be associated or linked to metadata. And that metadata is ideally readable for both humans and machines, and especially for machines, that gets important when it's about automating the data retrieval. So once the user has found the data, they need to know how to access the data. And this includes any kind of authorization and authentication, but also the machine needs to be supplied with a defined protocol, so to know how to do it. Then the next step in order to reuse data would, or the requirement would be for the data to be interoperable. And that is actually happening on two levels. So first of all, it needs to be interoperable with other kind of data, because most likely I would like to integrate my data with other data to do some meta-analysis. But it also needs to be interoperable with all kinds of machines or tools that we're using to process the data, to analyze it, or to store it. And then last but not least, the biggest step towards reusability is to supply the data with plenty of metadata, so that another user that is retrieving my data can actually make sense of the data, understand it, and also, for example, identify where are the limitations of the data. So the use case I'm presenting you today is still from my time when I was a PhD. So thanks again for the LICE lab for supplying me with this data and make use of it today. And it's data from a low throughput toxicological assay where we would cell neurite outgrowth and cell viability in response to compound treatment. And we would do that using 96 well plates, and we would have a microscope that is automated. But yeah, as soon as we would have the numbers and have the images quantified, we would use these structured, colorful Excel tables. And next to the actual raw data, we would even start doing some analysis, like a normalization. The compound information, so the information about which compound has been tested and also the concentrations would be even linked into further Excel sheets. And to unify this, I created, sorry, the clicker did not do what it's supposed to. There we go. I created this nice Knime workflow. So just quickly covering that again, before we would have like 50 different Excel sheets, 48 of them would have this colorful scheme, and we would have 2 more Excel sheets with the compound information and the concentration information. And that is really just, the information is just scattered around in different Excel files. And now with this Knime workflow, I would end up with 2 really nicely structured tables that are not only friendly for humans, but also for machines. And that would also be nicely enriched with metadata. So the first step to do this is actually to restructure the data. So when you recall how the data looked before, we had these 3 large blocks that are corresponding to the technical replicates, and the blocks that are in the row that are corresponding to the different endpoints. And you can also see we more or less kept the layout of the 96 well plate. And this is really nice for a human being to read, but it's not exactly machine friendly. So in the first step, I would use a loop to read in all the different data and then restructure it so we have a nice machine-friendly table where actually each row is corresponding to 1 data point. Then in the second step, I would merge the compound information. So I was already telling you, we had these 2 different Excel files. One of them contained the information about which compound was tested, and the other file would contain the information about the concentration. and they would only be linked to the actual data with the file name. So I joined this now to my data table. So this is where I ended up with, where I have a nice, concise data table where the information is just in there, and each actual number, each value is really linked to a concentration and a compound. With the third step, I was aiming at increasing interoperability. So initially, the data only came with CAS numbers, and this can be sufficient for some purposes, but for some it's not. So I extended the chemical identifiers with SMILES and INCHIs and INCHI keys. And also to make the data linkable, that is retrieved from other, for example, really large data sources like Campbell or Cabby. I was also enriching my data table with a controlled vocabulary from the Campbell and Cabby ontology. Nice side effect was that now also the human immediately gets what is actually the target of this compounds and what's its effect. Then with the fourth step, I was targeting the principle of reusability. And I did that by introducing a second data table. And I did that using the interactive table editor. So the user would get the possibility to describe the data that it's in each of the columns. And just make sure that anyone who gets a hand on this data really understands it, give more information, also link it to publications or protocols and things like this. And then in the last step, I would write the data. And before that, I was, again, still introducing an identifier, so a clear identifier within the data set for each data point. And I also introduced a kind of automatism so that the data, the file name that the user choose for the data file would automatically also define the file name of the metadata file so that these two are then clearly linkable. At that point, it would also be possible to write the data automatically from within Knime to a data repository, like, for example, Zenodo or Figshare via REST API. But this was not applicable for our project at that time. Then just to wrap things up, why Knime was a great tool to do this task. First of all, a big disclaimer. So I don't say that using Knime automatically would make any data fair. There are definitely principles that Knime is not really suitable for, that is mostly concerning findability and accessibility. But especially for the other two principles, interoperability and reusability, it's doing a really good job. First of all, because it can handle a large amount of data, and it's got it can really deal with many different file formats, and it can do all of that with any need of coding. And when I recall how we did it before, so it was really one PhD sitting there one day, copy pasting from one Excel file to the other, bringing it to a certain format that was required for this kind of project. And yeah, using Knime just really enabled us to release or free up this capacity in the lab and automate this kind of routine task. And with that, I'm already at the end and would ask Janina if we have any questions from the audience.
Yes, there's a very interesting question. Is FAIR also applicable to images?
Yes. Well, images are just data like anything else. So actually, it also applies. I would say it also applies to images. It's just at this point of the project, we were also integrating in data from other labs, and we just decided not to go and supply the images, but start from the quantification and yeah, just supply, so to say, the numbers and not the images.
Cool. Thank you.